Let us discuss the human blood groups. The human blood grouping system is known as ABO blood grouping system, and this ABO blood grouping system was given by a prominent scientist, that is Karl Landsteiner, right? His name was Karl Landsteiner. So Karl Landsteiner, he actually gave 3 types of blood groups, that is A, B and O. The 4th type of blood group, that is AB, it was given by D. Castillo and Steiner. Right? It was given by D. Castillo and Steiner. Now when we talk about the blood grouping system, then this blood group, it is determined by a very special gene, that is I gene. What is the name of the gene? It is I gene, that stands for iso-agglutinin. It stands for what? It stands for iso-agglutinin. Right? Now this I gene, it has 3 alleles. The 3 alleles are IA, IB and IO. So this I gene has 3 alleles, that is IA, IB and IO. Now IA and IB, these alleles, they are the dominant ones, means they are going to express. But this IO, it is recessive, means it is not going to express in the presence of a dominant one. So for the expression of a recessive one, you need to have both the combinations together, means you need to have IO and IO so that the blood group can be O. So what happens when you talk about the genotype? When you talk about blood group A, then the genotype is going to be IA, right? Or it can also be IA, IO. Okay. Why it is so? It is so because in this case, you can just make out, because in this case you both have IA and IA, then definitely the blood group is going to be A. But in this case, it is IA, IO. So IO, it is the recessive one, it is not going to express. Since we already have a dominant allele over here, then this is going to express, means the blood group is going to be blood group, that is A. Next, for blood group B, we have a genotype, that is definitely again IB, or it can be IB, IO. Again here this IO is the recessive one, so blood group B is going to be expressed, so the blood group of the organism is going to be B. Then we have AB. Now in case of AB, we just have a combination, that is IA, IB, right? Then in case of blood group O, we have the genotype, that is IO, and the next one, it is again going to be IO, because I told you that this is the recessive case, this is the recessive allele. So it is going to be expressed in just this case only when we have both the alleles that are recessive. If both the alleles are not going to be recessive, then this is not going to be expressed. So in case of blood group O, we have this genotype only, we have just this combination. Then these alleles, they are going to form the proteins. That proteins are known as antigens. What are those proteins known as? They are known as antigens. And these antigens, they lie on the surface of the RBCs. Where do they lie? They lie on the surface of RBCs. So what is going to happen? The organism who has blood group A, right, is going to have which antigen? A antigen, right? So blood group A person is going to have antigen, that is A. If we talk about the person having blood group B, then he or she is going to have antigen, that is B. If we talk about the person having blood group AB, then he or she is going to have antigen, that is both A and B. But if we talk about the blood group O, then they don't have any antigen. They neither have A nor do they have B. So there are no antigens, means none, right? Now if we talk about antibodies, then the opposite of this you will have to write here, and you will just use lowercase letter in this case. So if we talk about blood group A, the antigen is A, the antibody produced will be B. Antibody produced will be B. And if we talk about blood group B and antigen, that is B, then antibody produced will be A. Antibody produced will be A, opposite to that of this antigen, remember this, and write the antibodies in lowercase letters and the antigens in uppercase letters. Then if we talk about the blood group AB, then in this case there will be no antibodies that are going to be there. So antibodies here are none. But in case of blood group O, we don't have any antigens, but definitely we have antibodies, that is both A and B, right? We have antibodies, that is A and B. Now the question arises, how can we actually test the blood group of a person? For that we have anti-A serum and anti-B serum, right? What we are going to do, we'll actually have the blood sample from the patient or from the person who, from where you want to determine the blood group. Then you're going to mix that blood with anti-serum A and anti-B serum. If it is going to show clotting, right, then we are able to determine what blood group we are talking about. See, for example, in this case, in case of blood group A, then we have mixed the blood with anti-A serum as well as with anti-B serum. If there is clotting in anti-A serum, or you can say better word, coagulation of the blood with anti-A serum, it means the blood group of the person is A, since it is not going to show coagulation in case of anti-B serum. Alright. So we have this available in the lab, this anti-A serum and anti-B serum. We take that serum and we're going to put what? We're going to put the blood sample, and if it is going to show coagulation with anti-A serum, it means the blood group is A. And remember, you also have to check whether it is showing coagulation with anti-B serum or not. If it is not showing coagulation with anti-B serum, means it is definitely blood group A. Then next case is for blood group B. Now in this case, same, what is going to happen? The coagulation will be with anti-B serum. The coagulation will be with anti-B serum, and there will be no coagulation in case of anti-A serum. So this also confirms that the blood group is what? It is B. Then in case of AB, what is going to happen? I know that you can make out, because in this case there will be coagulation in both the cases with anti-A serum also and anti-B serum also. So if there is coagulation in both the serums, anti-A and anti-B, then it means definitely this blood group is AB, right? Remember, in case of A there was coagulation with only A. In case of B, there was coagulation with only B, but in case of AB, there was coagulation with both the anti-A and anti-B serums. So that is why I am just telling you, when you are going to check the blood group, you need to consider both the serums, right? Then in case of O, since it has no antigens, right, we wrote that it's none. So in this case, there will be no coagulation in any case. There will be no coagulation in anti-A serum and no coagulation in anti-B serum. Right. So this is how we actually check the blood group of a person. Right. Now coming to the question, what type of blood group can donate its blood to what type of a person, a different blood group, right? See, the person having blood group A can give blood to person having blood group A, and can get blood from what? From A, definitely, right? And then from blood group O also. Why? Because O is actually a universal donor. Then in case of blood group B can give blood to B and can get from B definitely and from O blood group as well, right? Now here I'll just like to mention that O is universal donor while AB is universal recipient. So in case of blood group AB, can get blood from A also, from B also and from O also, because AB is what? It is universal recipient, right? And can give blood to AB. Then in case of O can give blood to all of them, A, B and AB, but can get blood from only O, right? Why? Because O is universal donor, but can get blood from only O, right? So just remember that AB is universal recipient, right? Just we have mentioned, right? And O is universal donor. The next factor, very important one, is the Rhesus factor. Now in some cases, in some persons, there is Rhesus positive blood. Rhesus positive blood means their blood, their RBCs have another antigen that we call as Rhesus antigen. What is that? That is actually Rhesus antigen. Now this Rhesus antigen was actually first of all discovered in Rhesus monkey, that is a common monkey you see. And this Rhesus antigen is actually discovered by Wiener and Landsteiner. Wiener and Landsteiner. So they discovered this Rh factor, this Rhesus factor, which means that there is this Rh antigen on the RBCs in some persons. Means we are going to say that if there is availability of Rh antigen, then the person is Rh positive, and if there is no availability of Rh antigen, means the person is Rh negative. Now what happens? If supposingly the blood transfusion is going to be there, right? What is going to happen? If the person, or you can say the patient is Rh negative and the blood from the donor is Rh positive. So what is going to happen? The Rh negative blood is there, it is going to get mixed with Rh positive blood. So there is going to be the production of antibodies, right? There will be production of antibodies. Now when the antibodies are going to be produced, then for the first time it is going to be okay. But for the second time again, if the donor is Rh positive and the recipient is Rh negative, then there will be some reaction that will go on, and this is going to kill the RBCs. So this can even lead to death second time, right? First time it may not affect the person, but second time it can lead to death. So same is the case in case of pregnancy. What happens in that case? If the mother is Rh negative and the child it is Rh positive, what is going to happen? For the first time, if the blood comes in contact, if the blood from the mother and the child they come in contact, there will be development of antibodies in the mother's blood. If the second time the mother is going to conceive and the blood again comes in contact, since the antibodies were developed for the first time, second time it is going to affect the child, and the child will suffer from severe anemia, and the disease can be fatal also. The disease that is known as HDN, right? It is HDN. HDN is the disease which can actually cause fatality also, right? Severe anemic condition, and this disease is actually also known as erythroblastosis fetalis. It is erythroblastosis fetalis, right? Why it is there in the child? Because of the mixing of the blood. If the mother is Rh negative and the child is Rh positive, well, the child is Rh positive because the father is Rh positive. Remember that. The child is Rh positive because the father is Rh positive. For the first time, remember, when the mother conceives for the first time, then there is no problem. Why? Because the mother's blood is going to form antibodies against that. But second time again, if the child is Rh positive, then this can be fatal also, which causes this disease, that is HDN you can say, or you can also call it as erythroblastosis fetalis, right? So this is a very severe disease in which the RBCs of the child can get killed and the child can suffer from severe anemia. So this was all about the Rh factor. So remember, we have the blood grouping system, that is ABO, and the Rh factor is also very important if you talk about in terms of pregnancy, right? So the doctors need to check out if the mother is Rh negative or Rh positive, and if the child is Rh positive or Rh negative, as this can lead to abnormality, this can lead to abortion, and this can lead to this severe disease, right? So remember that these are the 4 human blood groups, and with this 4 human blood groups, you also need to remember about the Rh factor, that is the Rhesus factor, which is actually Rhesus antigen which is there in case of some persons. If there is Rhesus antigen means the person is Rh positive, if there is no Rhesus antigen means the person is Rh